The Mendelian genetic and chromosome. Do you ever wonder why you look like your brother, sister, where you got your cycle from? Are you concerned about developing a disease another family member struggles with? These are types of questions that we can answer with an understanding of genetics. We learn about the basic principle of inheritance and how likely it's to pass on certain traits from one generation to another. Pattern of inheritance. Many consider Gregory Mendel to be the father of genetics because his experiments in simple genetics provide foundation for modern genetics. Review Mendel law. Be sure to spend some time reviewing his figures. Genetics is the study of inheritance, hereditary. George Mendel set the framework for genetics long before chromosome or gene had been identified. At a time when meiosis was not well understood, Mendel selected a simple biological system and conducted methodological quantitative analysis using large sample size. Mendel's work considered the fundamental principle of heredity were revealed. We know now that genes carried on chromosomes and they are the basic function units of heredity with the ability to be replicated, expressed, and mutated. Today, the postulates put forth by Mendel form the basis of classical or Mendelian genetics. Not all genes are transmitted from parent to offspring according to Mendelian genetics, but Mendel's experiments serve as an excellent starting point for thinking about inheritance. George Mendel, who lived between 1822 and 1884, was lifelong learner, teacher, scientist, man of faith. As young adult, he joined the Augustinian Abbey of St. Thomas in Brno, in what is now the Czech Republic, supported by the monastery. He taught physics, botany, natural science at the secondary and university level. In 1856, he began a decade-long research involving inheritance patterns in honeybees and plants, ultimately settling on bee plants as his primary model system, a system with convenient characteristics that used to study the specific biological phenomena to gain understanding to apply to other systems. In 1865, Mendel represented the result of his experiment with nearly 30,000 bee plants to the local Natural History Society. He demonstrated that traits are transmitted faithfully from parent to offspring in a specific pattern. In 1866, he published his work, Experiment in Plant Hybridization, in the Proceedings of the Natural History of Science, in Bern. Mendel's work went virtually unnoticed by the scientific community, which incorrectly believed that the process of inheritance involves a blending of parent traits that produce an intermediate physical appearance in offspring. This hypothetical process appears to be correct because of what we know now as continuous variation. Continuous variation is a range of small differences we see among individuals in the characteristics like human height. It does appear that offspring are a blend of their parent traits when we look at characteristics that exhibit continuous variation. Mendel worked instead with a trait that show discontinuous variation. Discontinuous variation is the variation seen among individuals when each individual shows one or two or very few easily distinguishable traits, such as violet or white flowers. Mendel's choice of these kinds of traits allowed him to see experimentally that the traits were not blended in the offspring as would have been expected at the time, but that they were inherited as distinct traits. In 1868, Mendel became abbot of the monastery and exchanged his scientific pursuit for his pastoral beauties. He was not recognized for his extraordinary scientific contribution during his life. In fact, it was not until 1890 that his work was discovered in 1900 and revitalized by scientists. Mendel process. Mendel's similar work was accomplished using the garden bee Bisma sativum to study inheritance. Is species natural self-fertilizer, meaning that pollen encompasses over within the same flower. The flower later remains sealed tightly until pollination is completed to prevent the pollination of the other plant. The result is highly inbreed or true breeding bee plants. These are plants that always produce offspring that look like the parents. To experiment with a true breeding bee plant, Mendel avoid the appearance of unexpected traits in offspring. The garden bee also grows to maturity within one season, meaning that several generations could be evaluated over a relatively short time. Finally, large quantities of garden bee could be cultivated simultaneously, allowing Mendel to conduct that. His result didn't come about simply by chance. Mendel performed hybridization, which involves mating two true breeding individuals that have different traits in the bee, which is naturally self-pollinating. This is done by manually transferring pollen from the another of a mature bee plant of one variety to stigma of a separate mature bee plant of second variety. By experimenting with true breeding bee plants, Mendel avoided the appearance of unexpected traits in offspring that might occur if the plant were not true breed. The garden bee also grows to maturity within one season, meaning that several generations could be evaluated over a relatively short time. Finally, large quantities can be produced. Plants used in first generation crosses were called bee and parent generation plant. Mendel collected the seeds produced by the bee plant that resulted from each cross and grow them the following season. These offspring will called the F1 or the first filial, daughter or son generation. Once Mendel examined the characteristics in F1 generation of plants, he allowed them to self-fertilize naturally, having collected and grew the seeds from the F1 plant to produce the F2 or second filial generation. Mendel experiment extended beyond the F2 generation to the F3 generation, F4 generation, so on, but it was the ratio of characteristics in B, F1 and F2 generations that were the most become the basis of Mendel postulate. Here the violet flower and white flower or hybridization. All hybrid progeny have violet flower. It's a dominant violet dominant self-fertilization cross between the true breeding bee. Violet and white appear in the first generation. The second generation there is a percentage violet flower and white flower 705 in violet flower and 224 in white flower, about 1 to 3. Garden bee characteristic reveal the basic hereditary. In his publication, Mendel reported the result of his crosses involves 7 different characteristics, each with 2 contrasting traits. A trait is defined as a variation in the physical appearance of a heritable characteristic. The characteristics include plant heat, seed texture, seed color, flower color, bee pod size, bee pod color, and flower position. For the characteristic of flower color, for example, the 2 contrasting traits were white versus the violet. To fully examine each characteristic, Mendel generated large number of 1F2 plants and report results from thousands of F2 plants. What result did Mendel find in his crosses of flower color? First, Mendel confirmed that he was using plants that spread true for white and violet flower color, irrespectively of the number of generations that Mendel examined. All self-cross offspring of the parent with white flower had white flower, and all self-crossed offspring of parent with violet flower had violet flower. Mendel confirmed that other than flower color, the bee plants were physically identical. This was an important check to make sure that the two varieties of bee plants only different with respect to one trait, the flower color. Once this validation was complete, Mendel applied the pollen from a plant with violet flower to the stigma of the plant with white flower. After gather and restoring the seeds that resulted from this cross, Mendel found that 70% of the F1 hybrid generation had violet flower. Conventional wisdom at that time would have predicted the hybrid flower to be pale violet, or for hybrid plants to have equal number of white and violet flowers. In other words, the contrasting parental traits were expected to blend in the offspring. Instead, Mendel's results demonstrate that white flower trait had completely disappeared in F1 generation. Importantly, Mendel didn't stop his experiment here. He allowed the F1 plants to self-fertilize and found that 705 plants in the F2 generation had violet flower and 224 had white flower. This was ratio 3.15 violet flower to 1 white flower, 3 to 1. Mendel transferred pollen from plants with violet flower to stigma of plants with white flower, and etc. He obtained approximately the same ratio, irrespective of which parent, male or female, contributed in this trait. This is called the reciprocal cross, a paired cross in which the receptive trait of the male and female in one cross become the respective trait of female and male in other cross. For the sixth characteristic, Mendel examined F1 and F2 generation behaved in the same way that they behaved for the flower color. One of the two traits would disappear completely from F1 generation, only to reappear in the F2 generation at a ratio of roughly here the round and the wrinkled, yellow and green, purple and white, inflated or constricted, yellow or green. Upon compiling his results from many thousands of plants, Mendel concluded that the characteristic could be divided into expressed and latent traits. He called these dominant and recessive traits, respectively. Dominant traits are those that are inherited unchangeable in hybridization. Recessive traits become latent and disappear in the offspring of hybridization. The recessive trait does, however, appear in the progeny of the hybrid offspring. An example of dominant trait is the violet colored flower trait. For this same characteristic, flower color, white color are a recessive trait. The fact that the recessive trait reappeared in the F2 generation means that the trait remained separated and were not blended in the plant of the F1 generation. The recessive trait does, however, appear in the progeny of the hybrid offspring, an example of dominant trait in the violet colored flower trait. For this same characteristic, flower color, white colored flower are a recessive trait. The fact that the recessive trait reappear in the F2 generation meant that the trait remained separate and were not blended in the plant of the F1 generation. Mendel proposed that this was because the plant possessed two copies of the trait for the flower color characteristic, that each parent transmitted one of their two copies to their offspring where they came together. Moreover, the physical observation of dominant trait could mean that the generation composition of the organism includes two dominant versions of the characteristic. or that it includes one dominant and one recessive version. The observation of this trait means that the organism lacked any dominant version of this characteristic. Thank you.